Hello everyone, my name is Cassandre Pyne and I am a master's student at the University of Guelph in Dr. Mandeville's lab. I'm here today to talk to you all about the genetic basis of sex determination in Catostomus suckers. Sex determination is the system that determines whether an individual will be male or female. Surprisingly, sex determination is quite variable for such a fundamental process that all organisms with sexual reproduction will undergo. Not only is it incredibly diverse across the tree of life, as you can see in this figure, but also in the underlying mechanisms. For example, there are separate sexes in hermaphrodism, as well as environmental or genetic sex determination. The genetic factors that contribute to sex determination can also vary from a single sex-determining region, like a sex chromosome, to a polygenic basis with multiple regions working together to determine sex. You can see that in fish, this variability is quite apparent, with multiple different systems of sex determination at play. So in fish, as I pointed out in the last slide, sex determination is variable. This figure shows the known sex determination in fish, and although it is showing many forms of sex determination, we still do not know everything. Not only is information lacking on the systems of sex determination in fish, but also the mechanisms. Researchers believe that this high amount of variation is due to the genetic factors that are involved in sex determination in fish being less conserved than in mammals and birds. Due to all of this diversity, it can be quite difficult to make conclusions about how it impacts evolutionary rates. Incompatible sex determination systems may lead to reproductive isolation, which can impact speciation rates. So, understanding sex determination in a system may provide information about whether it promotes or impedes reproductive isolation. A family of fish that lends itself well to answering these types of questions are Catostomus suckers, which are widespread freshwater fish that are very species rich. They've diversified from a hybridization event around 50 million years ago, and hybridization is still ongoing in these fish. In addition, the outcomes of hybridization events are variable, and hybridization can be among native or introduced species. The figure on the right illustrates this. Each circle represents a hybridization event between the flannelmouth and the white sucker, which are pictured on the left, where each color corresponds to the degree of backcrossing between these two species, and the number inside corresponds to the number of hybrid combinations between up to 6 different species. So you can see that the level of hybridization varies geographically. The mechanisms that drive this variability are unknown, but genomic factors may be promoting reproductive isolation. Specifically, incompatible sex determination systems may be an effective reproductive barrier. So we can investigate any potential influence of sex determination on reproductive isolation and hybridization outcomes in Catostomus suckers. Although sex determination is undescribed in these fish, it is thought that sex determination has a genetic basis because other related fish have some form of genetic sex determination. The overarching research objective of this work is to identify the genetic basis of sex determination in Catostomus fishes. This will lead the way in identifying any potential influence of sex determination on reproductive isolation in these fish. We will also be investigating the basis of sex determination in each of the species as well, to determine to what extent the genetic basis of sex determination varies across species. The figure on the right illustrates 3 potential outcomes of this work, using a Manhattan plot, where the x-axis represents region of the genome, and the y represents how associated the region is to the trait of interest, which in this case is sex. Please note that the chromosomes and values are all arbitrary, as this is just an example. The dotted line shows the significance threshold, where any region above the line would be more associated with sex. So in A, we can see that one region is above the significance threshold, so this would indicate that sex determination is under single locus control, most likely a sex chromosome or a master sex determining region. In B, no regions reach the significance threshold. This could indicate that sex determination is either super polygenic, meaning that it is under control of so many loci of small effect that it is not picked up, or that there was nothing there. In C, there are multiple regions associating with sex, so this situation would represent a polygenic basis of sex determination, where multiple loci work together to determine sex. Any of these outcomes would be informative because they would provide information about how sex is determined in these fish. So to answer the previous question, I am using genomic data to determine regions of the genome that are associated with sex. We have samples from Colorado of 3 different species of Catostomus fishes, bluehead, white, and flannelmouth, as well as their hybrids. We currently have about 100 samples, but just recently doubled this data set as we just received data back from the sequencing facility. In addition, we have a reference genome for the flannelmouth sucker that will be used for this analysis. I've completed the DNA extractions on the fin clips and sent the samples off for sequencing, specifically reduced representation sequencing. I then completed bioinformatics work to clean the data and prep it for analysis. And the analysis consists of determining regions of the genome that are associated with sex in just one sex, as well as identifying sex-associated loci in both sexes. And I'll go into a bit more depth about the analysis in the next few slides. So the first part of the analysis is determining regions of the genome that are associated with sex in both sexes. So this could be consistent with homomorphic sex chromosomes or a polygenic basis. The figure on the right illustrates this method. The two genomes shown are of a male and a female, and each have a region that associates with sex that maps to the Manhattan plot. In this example, there's just one significant peak, so this could be a sex chromosome or master sex determining region. However, it is important to note that this figure can be reproduced with multiple different regions as well. For this project, I am using a genome-wide association study, specifically the software GWAS. This software uses a linear mixed model to detect associations. The second part of my analysis is to determine regions of the genome that associate in just one sex but not the other. So this would be sex-specific loci, which is consistent with heteromorphic sex chromosomes. The figure on the right again illustrates this method. So it involves analyzing patterns of missing regions. And you can see that in A, the male genome is missing a region that the female has. So this may be sex-specific and would most likely be female heterogamity in this case. The opposite is the case in B, where the female is missing the region. It is important to analyze patterns of missingness because a simple presence-absence scan may not take into consideration differences in sequence coverage. So basically, this method is trying to answer the question of whether these loci are really sex-specific or if they're just missing by chance. So a statistical threshold is then used to determine the amount of missing data necessary for it to be considered statistically sex-specific. So I wrote a custom JavaScript to complete this part of the analysis, which is currently in its finishing stages. So far, we have conducted a genome-wide association study with the samples that we currently have. Here are the preliminary results using GWAS. So again, on the x-axis, this is the scaffold. So that's region of the genome. And on the y is the negative log of the p-value. But you can just think of it as a measure of how much each loci is associated with the trait of interest, which again is sex. You can see that the blue indicates a peak at scaffold 1, 2, 3, 4, 3. And this is a super exciting result because it is indicating that there's a region of the genome that is associating with sex, and it really looks like it could be a sex chromosome or a master sex determining region. This again is just from running one software with a sample size of 100, but it is exciting nonetheless. So to investigate this a bit more, I plotted just that chromosome to see if the significant region is still clustered together. So this is the result of plotting just that one significant scaffold. You can see that the significant region is clustered together on the scaffold, again, looking like it is one specific region that is hitting the significance threshold. These results are indicating that the genetic basis of sex determination in suckers is under single region control, at least so far as this work is still ongoing. The other method for my project is the identification of sex-specific loci. I won't go into too much depth about the specifics of the program, but as I mentioned before, it is a Java program that analyzes patterns of missingness across the genome between males and females. It identifies the sex-specific loci in the simulated data, and it's currently in the testing phase, meaning that we are using data sets with known sex determination. So it will most likely undergo tons more edits before it's fully finished. However, I did test it on a filtered Catostomus sucker dataset, but it did not detect any sex-specific loci. So, from the preliminary results, I can start to make a few conclusions. First, using the potential outcomes that I presented earlier, we can start to eliminate possibilities. It currently does not look like scenario B, where no regions hit the significance threshold. However, it may be A, where there is one significant region, but it also could be C. This is because when we split the dataset by species and their hybrids and start to investigate how they vary, we may start to see other regions hitting the significance threshold in different species. So the one significant region that we are seeing in our results right now may just be the one part that is shared among all species. So this may just be the shared basis of sex determination. So this one region may be the region large enough to show up in the analysis, where smaller regions may be significant in just one or two different species. If sex determination in Catostomus fishes is a single sex determining region, the question becomes, how do we think this contributes to reproductive isolation? A single sex determining region that is shared among all species may allow for another system or factor to contribute to hybridization outcomes in these fish. On the other hand, if it becomes more apparent that it is polygenic, this may mean that there is possibility for more incompatibilities between species if there is variability in the loci contributing to sex determination. So although we have preliminary results, there are still questions that are waiting to be answered as this project continues. Overall, this is very exciting because even with all of the species added together in this analysis, with only the first part of our samples, and with data that only targets a small proportion of the genome, we still found something. So, as mentioned previously, this work is still ongoing. First, we received the next data set of about 100 samples, so we can rerun the genome-wide association study, as well as split the data by species, and I can start to answer some of the questions that I outlined in the previous slide. We will also be continuing with the testing of the Sexspecific loci program. We'll be using datasets with known sex determination to make sure that it actually works. We also have a very similar dataset for 2,700 individuals of unknown sex, so we will be able to test these methods on those fish. If we believe that we found a sex-determining region in Catostomus suckers, we can test it by identifying the 2,700 individuals. This will also be especially helpful in testing the method that we are developing for the identification of sex-specific loci. I would like to conclude today by giving a few take-home messages. Overall, this research will add to our understanding of biodiversity in Catostomus suckers. By identifying the genetic basis of sex determination in suckers, we can start to understand how reproductive isolation occurs and varies. Specifically, we can start to understand the extent that reproductive isolation is impacted by incompatible sex determination systems. From a conservation standpoint, this work will be especially useful in the field. It can prevent the death of fish in the field when sex information is needed, because we would be able to determine the sex from DNA from the fin clips. In addition, this research can have some conservation relevance because some suckers, specifically flannelmouth and bluehead sucker, are of conservation concern. All right, so that is the end of my talk today, but first I would like to thank NSERC, Colorado Parks and Wildlife, and my home department of integrative biology at the University of Guelph. I would also like to thank the Mandeville lab for all of their comments and assistance. Thank you for listening.